Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad MSc Entrance Examination question paper for the subject Plant Biology and Biotechnology conducted in the year 2021, and I will be discussing only the biology part. If you are new to my channel, please like, share and subscribe. First, coming to the first question. An instrument used to measure the amount of water vapor in air, in soil or in confined spaces is called. It is called as the hygrometer. Answer is option B. Then, one of the research scholars has isolated a novel protein from the medicinal plant which enhances the immunity in humans. After isolation, he purified the protein by a protein purification method in which he used Cephedex G100. Which technique he has used for the purification of novel protein? So he has used gel filtration chromatography. Why? Because Cephedex G100 is used in gel chromatographic techniques. A similar question was asked for one of the entrance examination conducted by the university. I don't remember which paper they had asked, but it was asked. Then, three polypeptides whose masses are 55, 50 and 75 kDa with pi of 6.5, 7 and 8 respectively were subjected to standard reducing SDS page. The order of their separation from top to bottom would be. So here SDS page, it separates the protein primarily on the basis of mass and not on the basis of pi. So SDS is an ionic detergent and it will bind to the protein to make them uniformly negatively charged. So the top, so they have asked from top to bottom. So the top one will be the one with maximum molecular weight. So it is C, that is 75 kDa. Then comes 55 kDa, that is A, and then comes C, that is then comes B, that is 50 kDa. So the order is C, A, B. Then next one. The scientist Brunner, Meyerhoff, Parnas and Embedden have made significant contributions in the field of. So it is in the field of respiration in plants that they had made significant. And Embedden, Meyerhoff, Parnas pathway is also called as glycolysis. Then which one of the following microscopes would you use to localize a protein fused to a green fluorescent protein reporter in a cell? So for viewing that you will use confocal microscope. Answer is C. Then a seed biologist tested a seed population for generation. In this tested population he got 68% germination, and out of 32% ungerminated seeds, 25% seeds were found viable. What type of analysis he might have performed? So he has checked for the viability of the germinated seed. So what test did he use? He used tetrasolium test. Answer is option A. In higher plants, tunica corpus theory is applicable to. It is applicable to the shoot apex. Answer is B. From a protein mixture, some molecular weight proteins can be separated by using. So, same molecular weight proteins can be separated by using 2D page. It is two-dimensional polyacrylamide gel electrophoresis. So, the 2D page, it separates proteins by native isoelectric point in the first dimension and by mass in the second dimension. So you can separate a mixture of same molecular weight proteins. So answer is option. Then next one. The degree of intensity with which a particular genotype is expressed in an individual is called as expressivity. Answer is option B. C55H72O5N4Mg is the chemical formula of chlorophyll A. Answer is option A. Then viviparous nature of seed germination occur in Rhizophora. This is also a repeated question. Then bioreactors are useful in. So they are useful in processing large volumes of culture. Answer is option B. Oxenometer is used for measuring of plant growth. It is used for measuring plant growth. Answer is option C. Then cultivated bananas are sterile because they are triploid and therefore seeds are not set. Answer is option C. Next one. Which of the following is incorrect for H plus ATPase? So it uses energy of hydrolysis of ATP. That is H plus ATPase uses energy of ATP to pump H positive protons out of the cytoplasm of the cell. Then it is also responsible for maintaining cytosolic pH in the range of 7.3 to 7.5. That is also correct. It results in the generation of protein motive force. So when the H positive ions or protons are pumped out of the cytoplasm of the cell, this creates and maintains an electrochemical gradient of H plus outside of the cell to inside of the cell. So that is also correct. Then it maintains a high H positive concentration inside the cell. It doesn't do that. So answer is option D. That is the incorrect about H plus ATPase. Then during ABA mediated stomatal closure, membrane depolarization is caused by net influx of which of the following ions? So it is hydrogen. Answer is option C. Then next one. How many number of chiral atoms are present in glucose and fructose? So this is the glucose and there are 1, 2, 3, 4 chiral atoms in glucose, and fructose it has got only 3 chiral atoms, 1, 2 and 3. So the answer here is 4 in glucose, 3 in fructose. Then next one. Mutations that involve the change of a single nucleotide and that code for different amino acids are. So they result in change in single nucleotide and they code for a different amino acid. That is the case of missense mutation. Answer is option A. Nonsense mutation is resulting in the termination codon, and transversions is a purine getting converted to pyrimidine or a pyrimidine getting converted to purine, and frame shift mutation is change in the normal reading frame. Then next one. Which of the following nutrients is required for nitrogen fixation in higher plants? So it is cobalt, CO. Answer is option C. Then during sexual reproduction in barley, how many total meiotic divisions will be required for forming 100 zygotes per 100 grains? So in plants, 4 male gametes are produced by a single microspore via meiosis. So after 25 divisions, 100 male gametes will be produced. Also, 1 female gamete is produced after a single meiotic division. Hence, 100 eggs will be produced in 100 division. Hence, a total of 125 meiotic divisions, that is 100 plus 25, are required for the formation of 100 zygotes per 100 grains. So the answer is option C, that is 125. Then the flavor saver tomato was the first genetically engineered crop product commercialized for human consumption. This transgenic tomato is known for enhanced shelf life. Answer is option C. Then cysts are commonly seen in Azotobacter. Answer is option D. Then identify the correct sequence of events that lead to the development of female gametophyte in angiosperms. So it is megaspore mother cell, MMC, meiosis, PCD, functional megaspore, gametogenesis, and then gametes. Answer is option B. Then next one. The carbon dioxide compensation point for C3 plants is greater than C4 plants because in C3 plants, photorespiration is present. Answer is option A. Then which of the following events take place in metaphase 1 of meiosis? So homologous pair of chromosome line up on the metaphase plate. In most of the question papers, I have seen one question from meiosis. That to metaphase 1 or anaphase 1 they will be asking. and also whether difference between meiosis 1 and meiosis 2, that is regarding separation of the chromosomes and chromatids, also they have asked. So be thorough with meiosis. Then next one. The phytochemical alvane is a potential inhibitor of. It is a potential inhibitor of Na plus K plus pump. Answer is option C. The first alkaloid to be isolated and characterized from plant is. So it is morphine. Answer is option B. Then kinase reaction involves. So it involves either the addition or removal of a phosphate group. Answer is option B. Then restriction endonuclease. Not one recognizes 8 nucleotide sequences in a genome. What is the probability of the presence of not one recognition sequence in the genome? So, there are how many? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleotide sequences for not one. And they have asked what is the probability of the presence of not one recognition sequence in the genome. So, what is the probability of finding a G at position 1? Now, this is position 1. So, what is the probability of finding G at position 1? That is over here. That is 1 by 1, 2, 3, 4, 5, 6, 7, 8. 1 by 8. Similarly, what is the, 1 by 4 it is, okay, because there are 4 nucleotides, so 1/4. Then C will be, that is also 1/4. This third position G also 1/4. Fifth position G also 1/4. Sixth position C is 1/4. Then this C is 1/4. So all are 1/4. So you will get total of 1 by 4. Okay. 1 by 4 raised to 8. So what is the probability of the presence of not one? So the probability of finding this particular sequence in the genome is 4 raised to 8 or 4 raised to n. You can directly use the formula 4 raised to n, that is 4 raised to 8. If such a question is asked, better go directly for the formula 4 raised to 8 or 4 raised to n. N, that is the formula. So 4 raised to 8, that will be 65,536 base pair in the genome. So it should cut after every 65,536 base pair, you will come across a not one sequence. After every 65,536, that is distance between two not one fragments or two not one recognition sequences is 65,536 base pair in the genome. That is 4 raised to 8. So the answer is option D. Now when you are learning, be thorough or just learn by heart what is 4 raised to 8, because I think you are not supposed to use calculators for your examination. So it is better if you could just memorize how much is 4 raised to 8, how much is 4 raised to 4, how much is 4 raised to 6, and how much is 4 raised to 3. Okay, 3 is rare, but 4, 6, 8 and 5 I have seen. So answer is option D. Then which of the following statement is incorrect in angiosperm? So they have dominant gametophytic generation, that is incorrect. Then double fertilization occur, that is correct. Presence of lignified vascular system, that is also correct. And independent sporophyte and gametophyte stages, that is also not correct. So answer is option A and D. Then dash products mRNA from degradation. So series of adenine bases, poly A tailing, out of these given options. So answer is option C. Then lateral roots originate from. So lateral roots, they originate from the pericycle cells lying against protoplasm. Answer is B. Then entry of enveloped viruses into its host cell is mediated by. So it is mediated by both endocytosis and membrane fusion. So answer is option C. The next one. Which of the following viruses is not reported to spread by fruit bats. So answer is HIV. Whereas SARS, Nipah and Ebola, they are spread by fruit bats. Then, on the sequence analysis of a double-stranded DNA, the result showed the content of guanine was 30%. What is the amount of A and T put together? So, G is 30%, then the DNA is double-stranded, so C also will be 30%. So, G plus C will be 60%. And they have asked how much will be A plus T. So, 100 minus 60, that is 40%. So, it is answer is option A, 40%. Now, if they have asked how much is A or how much is T, then it will be A will be half of 40, that is 20%, and similarly T also will be half of 40, so it will be 20%. Then, identify the correct statement regarding red-green color blindness which is caused by the presence of an X-linked recessive allele. So, it is an X-linked recessive allele, so it is located on the X chromosome. X-linked recessive genes are expressed in females only if there are 2 copies of the gene. However, males, there need to be only 1 copy of an X-linked recessive gene for the trait to be expressed. So here, if you see option A, 2 colorblind parents can give birth to a normal son. So X raised to C that I have written as colorblind parent. So female is also colorblind and the male is also colorblind. Can give birth to normal son. So when you see the cross over here, only colorblind son and colorblind daughter are present. So they cannot give rise to a normal son. Then 2 colorblind parents can give birth to a normal daughter. Here we can see that only colorblind daughter is present. So they can give rise to only colorblind daughter, not to a normal daughter. Then 2 normal parents can produce a colorblind daughter. So these are the 2 normal parents. So female will be either heterozygous or will not be a carrier of that particular gene. So males are normal, so they will be XY only. So if the female is heterozygous, then she will produce 2 types of gametes, and upon crossing you will get no colorblind daughter but only carrier daughter. So 2 normal parents can produce colorblind daughter. So that is also wrong. Then if the female is just XX, then she will produce only normal son and normal daughter. Then 2 normal parents can produce a colorblind son. So in that case, 2 normal parents means one will be heterozygous and the other will be normal. That is, the male will be normal and the female will be heterozygous. In that case, you can chances of getting a colorblind son is there. See over here, you can see that colorblind son is produced. So answer is option D. That is correct statement regarding red-green color blindness. Then next one. Match the common names of the species listed in column A with their scientific names and the plant parts commonly used in food preparations given in column B and column C respectively, and choose the correct answer. So cloves. Cloves is Syzygium aromaticum and it is flower buds. Then next one, black pepper. Black pepper column B, you will find it is Piper nigrum and the part used is fruits. 2, 4. Then 3, cardamom. Elateria cardamomum, that is D, and seeds are used. 1. And turmeric 4, it is curcuma longa and so it is the B, and part that is used is roots. So 2. So the answer is option A, that is 1 C3, 2 A4, 3 D1, 4 B2. Then next one. If seeds become dormant when present on a mother plant itself, such type of dormancy is known as, innate dormancy. Answer is B. The process of copying RNA into DNA is called as reverse transcription. Answer is option B. Then Gothe's foliar theory, Hogan and Somerville's ABC models relate to the study of flower development. ABC model, it is in Arabidopsis. So answer is option D, that is flower development. Then which plant is the source of natural alternative to hydroxychloroquine antimalarial drug? It is cinchona bark. Answer is option C. Seed germination is completed when the primary root comes out of the seed. Answer is option C. Then double fertilization it occurs in. So double fertilization occurs in angiosperms. Answer is option C. Then name the products of beta oxidation of fatty acid. So it is acetyl-CoA and FADH2 and NADPH2. So the option C and D both are correct. Then according to the second law of thermodynamics, spontaneous reaction will occur when molecules having lesser entropy are converted to molecules having higher entropy. Answer is option D. Excited state of chlorophyll molecule responsible for the photochemical reaction of photosynthesis is. It is the first singlet state. Answer is option A. Then next one. Stomatal closure is induced by. It is induced by abscisic acid. Answer is option A. Which of the following is not an auxin transport inhibitor? TIBA, NPA, morphactin, NAA. NAA is auxin itself. That is naphthalene acetic acid. How can auxin itself inhibit auxin transport? So the answer is NAA. Then which of the following is a natural growth inhibitor which has physiological properties similar to ABA? So it is xanthoxine. Answer is option A. Then any partial unfolding of the protein destabilizes the protein structure. In such circumstances, which form of the secondary structure components can be seen? So you can see random coils. Answer is option D. Then output of glycolysis if one glucose molecule metabolizes. So it is 2-pyruvate, 2-ATP and 2-NADH. Answer is option A. Then proteins having KDEL, that is lysine, aspartic acid, glutamic acid, leucine signal sequences are. So they are endoplasmic lumen retaining proteins. Answer is option A. Then the three types of C4 plants differ from each other on the basis of. So they differ on decarboxylation reaction of C4 compound in the bundle sheath cell. Answer is B. Then which of the following species produce non-endospermous seed? So it is Phaseolus vulgaris. Answer is C. Then termites represent, it is tripartite biological system. Answer is option C. Then read the following statements regarding chelating agent and identify the correct statement. So correct statement is these are organic compounds which are having the ability to form stable water soluble complexes with heavy metal and prevent or reverse the binding of metallic cation to ligands, improving the metal's general stability and likelihood to bond with other substances. Answer is option D. Then phycoerythrin and phycocyanin are the pigments commonly present among the members of the phylum Cyanobacteria. Chemically these are, so they are open chain tetrapyrols coupled to proteins. Answer is option B. Then Clostridia which obtain energy by fermenting amino acids in which one amino acid acts as electron donor and is oxidized, whereas the other acts as the electron acceptor and is reduced. This type of coupled decomposition is known as. So it is Stickland reaction. A similar question was asked previously in 2 question papers. 2 years. Okay, then a geneticist interested in immune function induces random mutations in a number of specific genes in mice and then determines which of the resulting mutant mice has impaired immune function. This approach is an example of. So this is an example of reverse genetics. Now before coming to that, what is reverse genetics? So it is an approach to understand the function of a gene. How first you will select a gene, then you generate a null mutant of the gene, then you study or analyze the phenotypic effects. What is forward genetics? forward genetics is you have the phenotype. So your method is to determine the genetic basis responsible for this particular phenotype. For that, what you will do, screen a collection of mutants, then identify the gene causing the phenotype. That is in the case of forward genetics. Now what have they told here? So in this question they have told that the genetist selected the gene for immune function. So the gene was selected first. Then random mutations were induced in a number of specific genes. Then he or she determined the phenotype, that is which function have impaired immune function, that was determined, that is the phenotype. So it is the case of reverse genetics. Then next one. These are multicellular green algae characterized by flattened growth form composed of thin-walled cells. These are thought to be sister groups of Charlie's plus land plants. Carrales plus land plants. These are coleochaetae. Answer is option A. Then position of centromeres can be determined by using C banding technique. Answer is option C. The anticodon of tRNA fmet is. So methionine, that is codon is AUG, that is given in the 5' 3' direction. Now anticodon you have to take from the 3' to 5' direction. So 3' to 5' will be UAC. Answer is option C, that is C. Then which of the following is or are true about isoenzymes? So they have several identical subunits. No. Then they react with the same substrate but form different products. No. Because glucokinase and hexokinase, if you see, they catalyze the reaction of glucose to glucose 6-phosphate. So they yield the same product. So that is also not correct. They are obtained from the same gene by different post-translational modification. So isoenzymes are coded by genes at different loci or are products of different genes. Okay. So third one is also wrong. Then fourth, they are proteins with different primary structure that perform the same reaction on the same substrate. So 4 is correct. Answer is D, that is 4 alone. Then trehalose is a disaccharide formed by 2 alpha glucose units. Answer is option A. Then circadian rhythm is a biological rhythm with a cycle of 24 hours. Answer is option B. Which of the following organic acid is present in rancid butter? So it is butyric acid. Answer is option D. Then the positive charge in NAD plus is found in which part of the molecule? So it is found on the nicotinamide part of the molecule. So answer is option A. Then Michaelis-Menten plot enzyme kinetics, reaction rate versus substrate concentration. The reaction rate reaches a plateau and does not increase any further upon adding more substrate. Is that all substrate have been converted to products? No, the active state is saturated with the substrate. That is the answer. That is option B. The substrate is inducing inhibition on the enzyme. No, there is competitive inhibitor present. No. So the answer is option B. The next one. Match the antibiotics in group 1 with the targets in group 2 and choose the right answer. Sulfonamide, folic acid biosynthesis. That is 1, 3. Then quinolones. Quinolones is topoisomerase. Erythromycin is peptide chain elongation. And cephalosporin is peptidoglycan synthesis. So the answer is 1, 3, 2, 4, 3, 2, 4, 1. Answer is option D. Then the diploid chromosome number of Datura stramonium, the Jimson weed, is 24. How many different types of trisomics can be obtained in this species? The haploid chromosome number is 12. So 12 different types of trisomics can be obtained in this species. Answer is option A. Next one. Mature male gametophyte in plant is, it is the pollen grains. Answer is option A. Then match the following and choose the correct option. Requirement of light for germination. It is photoblasty. Folding of leaves at night. It is nyctinasty. Opening of leaves at dawn. That is photonasty. And directional bending of shoot to light. That is phototropism. So answer is option C. That is 14213243. Then quantosomes they are found in thylakoid membrane of chloroplast. Answer is option A. Then what is the value of delta G when the reaction is at equilibrium? It is delta G is equal to 0. Lineweaver-Burke plot is also known as double reciprocal plot. Answer is option B. Which of the following is not a diazotrope? So diazotrope are bacteria and archaea that fix atmospheric nitrogen gas into more usable forms such as ammonia. So here Clostridium botulinum is not a diazotrope. So answer is option D. Then in cereal, the cells of the outermost layer of the endosperm become morphologically and physiologically specialized to form a layer called as aleuron. Answer is option A. These are all just direct questions. Okay, that's why I am not explaining much. Virtually all bacteria have cell walls containing peptidoglycan. Which among the following is an exception? So chlamydia is an exception. Then which among the following organism is recognized as the causative for the 7th pandemic during the early 1960s? So it is E1T or biotype of Vibrio cholerae. Answer is option B. Then in the negative regulation of gene expression, the presence of a particular regulatory factor reduces gene expression and its absence result in increased gene expression. The factor is repressor. If you see the case of lac operon, so the presence of this particular regulatory factor, that is this particular repressor, it reduces gene expression. So if you see in case of lac operon, the repressor will go and bind to the operator. It will prevent the RNA polymerase from binding to the promoter and resulting in transcription and translation of no transcription and translation of the genes responsible for lactose metabolism. Then its absence results in increased gene expression. Now when the repressor is not present or when repressor is bound to lactose, what will happen is it will result in repressor not able to bind to the operator. So RNA polymerase, it will be able to bind to the promoter and will result in transcription and translation of lactose metabolizing genes. So the answer is option B, that is repressor. Which enzyme is involved in epigenetic inheritance? So from here it is histone methyl transferase from the option. So epigenetic changes alter the physical structure of DNA. It can be either through DNA methylation, that is addition of a methyl group, or a chemical cap to part of the DNA molecule which prevents certain genes from being expressed. And another is through histone metabolism using or histone modification using histone methyl transferases. So answer is option D. Then which of the following pair is interconverted in the process of mutarotation? So it is alpha D glucose and beta D glucose. Answer is option A. Next one. Which element plays an important role in pollen germination? So it is boron. Answer is option D. Usually in photosynthesis, the resonance energy plays a major role. Therefore, what is resonance energy transfer? The transfer of energy but not the electrons. Answer is option B. Then, in general, the melting point of fatty acid depend upon chain length and also on the degree of unsaturation. Answer is option D. So, melting point of unsaturated fatty acid decreases with increase in double bonds. Then, as the number of carbons in fatty acid chain increases, so does the melting point also. Then compounds derived from burnt plant material responsible for stimulating seed germination in plants they are carychins. Answer is option D. Then which precursor is involved in biosynthesis of folate pathway? So it is shikmate. Answer is option B. Next one. A reciprocal cross was made between two types of the evening primrose. Enothera hookeri and Enothera muricata, known to have the same chromosome constitution. When the seed parent was Enothera hookeri, the plastids of the progeny were yellow. But when the seed parent was Enothera muricata, the plastid of the progeny were green. Which of the following statement is the correct explanation of the differences in result obtained from the reciprocal cross? So, one minute. So what is reciprocal cross? It is a cross between male of one strain and a female of another. Or else it can be the female of one strain and the male of the another, or vice versa. So here when the seed parent, that is the female parent, was Enothera hookeri, the plastids of the progeny were yellow. So all were found to be yellow colored plastids. But when the seed parent, that is the female parent, was Enothera muricata, the plastids of the progeny were all green. So what is the correct explanation? So it clearly shows maternal inheritance. That is, Enothera hookeri contains yellow plastids. So when that was used as the seed parent, all the progeny had yellow plastids. And when Enothera muricata was used as the seed parent, all the progeny had green plastids. So it is a typical case of maternal inheritance. So answer is option C. Then porins are. So porins are present in the outer membrane of gram-negative bacteria and they function as channels for the entrance and exit of hydrophilic low molecular weight substances. Answer is option A. Then, the molecular markers and their classification are given below. Which of the above combination are correct? So, SSR and is codominant. RFLP is also codominant. So, one is correct. Then ISSR, it is dominant. And RAPD also it is dominant. So, they are not codominant. Then AFLP and RAPD both are dominant, not codominant. Then AFLP and IISR are both dominant. So answer D is also correct. So answer is option D, that is 1 and 4 are correct. Then next one. Which of the following is an anaplerotic reaction? So anaplerotic reaction is the anabolic reduction that helps to generate the intermediate compounds of the biochemical pathway. So here conversion of pyruvate to oxaloacetate is an example of anaplerotic reaction. Answer is option D. Then a type of facultative apomixis in which the male gamete does not fuse with the female nucleus following entry into the embryo sac lead to the production of paternal and maternal haploids is. So it is called as semigamy. Answer is option A. Then the prophase 1 of first meiotic division is characterized by critical events as reflected by the appearance of chromosome at each substage. The stage of prophase 1 during which synaptonemal complex dissolves. So crossing over has already taken place. And the pair of homologous chromosomes remain held together at intervals by cross connection resulting from crossing over. So they have asked for which stage synaptonemal complex dissolved and the pair of homologous chromosomes they remain held together at intervals by cross connections resulting from crossing over. So the way sorry sorry. So the stages of meiotic prophase is divided into the meiotic prophase 1 is divided into 5 stages. That is leptotene, zygotene, packetine, diplotene and diakinesis. So in this they have asked where the synaptonymal complex dissolves. That is crossing over has taken place. So it is not neither leptotene, zygotene or packetine. So it must be diplotene. So answer is option D. And one other option also they have given, that is the pair of homologous chromosomes, they remain held together at intervals where by cross connections resulting from crossing over. So these intervals, that is where there is cross connections, they have resulted after crossing over. So this stage is called as the diplotene stage. Then next one, which of the following statement is incorrect about somatic embryogenesis in vitro? So somatic embryos have vascular connections with the original tissue. That is not correct. Answer is option C. Then next one, the antibiotic fusidic acid inhibits the elongation step of protein synthesis. Which other antibiotics have the same mode of action? So it is virginomycin and pulvomycin. Answer is option C. Then, in a series of 3 alleles is known to exist in a given diploid species, how many different combinations might be expected to occur in the diploid individual of the entire population? So in this case, if you take the example of ABO blood group, there are 3 alleles for blood groups and you can get 6 different phenotypes. So the expected number of expected number of different combinations that can occur in the diploid individuals of the entire population will be 6. Answer is option B, that is 6, 97B. Then, citral is used extensively in the perfume and flavor industry to stimulate lemon-like odor. Upon oxidation, it gives rise to geranic acid. Answer is option B. Then in siamese, the diploid chromosome number is 20. The chromosome number and ploidy of mature endosperm. So the endosperm will be 3N and N will be 20 divided by 2, so it will be 10. So 3N will be 3 into 10, that will be 30 because endosperm is triploid. Answer is option C. Then which of the following is not an allopolyploid? So out of these, Solanum tuberosum, it is not an allopolyploid. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.